Sorry I couldn't make it to Sweden. Unfortunately, I'm snowed in Mongolia. I'll be doing my presentation on Parkinson's disease, which is a progressive, long-term genetic condition of the central nervous system, with it predominantly affecting the movement of a person that is caused by changes on the DNA information and human chromosomes. This neurological disorder is linked to the decline in the production of dopamine, an important chemical of the brain that is classified as a neurotransmitter. This decline of this chemical subsequently leads to the lack of control people have over the movement of their limbs in their body. There are a number of physical symptoms that people with Parkinson's disease have to deal with on a day-to-day basis. The symptoms associated with this genetic condition get progressively worse over time. Early symptoms may go unnoticed as they are generally quite mild, with signs of having this condition usually beginning on one side of the body and then spreading. The main physical symptoms include traumas, muscle stiffness, increased slowed movement, disrupted sleep, balance and posture problems, and a decline in the ability to carry out automated movements. The genes responsible for this genetic condition are found within two human chromosomes, identified as the Parkin gene, located on chromosome 6 on its long arm, and the alpha-syncline gene, located in chromosome 4 on its long arm. The changes that occur in the DNA of patients with this condition includes a genetic mutation in the dopamine-producing neurons of the brain. In common cases of Parkinson's, a protein known as Lewy bodies appears in these neurons that disrupt the smooth transition of signals within the brain. These mutations seem to impact a component of the cell that is responsible for breaking down unwanted proteins, that ultimately leads to the buildup of these proteins within these neurons, eventually causing them to die. Other mutations include effects on the energy-producing components of the cell, known as mitochondria. In only 15% of cases is Parkinson's found to be hereditary, running in the patient's family history. This genetic condition can be either autosomal dominant or autosomal recessive, depending on the gene that is altered. In cases of being dominant, we see the SNCA or LRRK2 genes follow a pattern where an affected person generally only has one parent with the condition, because one duplicate of the altered gene is enough to trigger this condition. Otherwise, in cases of being recessive, we see either the PRKN, PARK7 or PINK1 genes follow a pattern where an affected person's parents both generally carry one copy of an altered gene, but don't display any signs or symptoms of the disease. Therefore, meaning two copies of the gene in each cell of the individual are changed. This is evident through this pedigree chart and this Punnett square that show the chances of inheriting this condition, where if it being dominant there is a 75% chance, or recessive a 25% chance. For this disease to be accurately diagnosed, it requires a trained neurologist to accurately provide an analysis of the signs and symptoms the patient has, and they'll make a diagnosis based on a number of differing factors, including a review of medical history, responses to tests, etc. They will then need to perform a physical examination, further supported by neurological testing, as there is no particular test to diagnose this condition. With no cure for Parkinson's, there are no current treatments to totally eliminate the condition and reverse its effects. Rather, there are treatments in managing the symptoms associated with it. Medication is used to increase levels of dopamine in the brain and control parts of the brain where dopamine is found. This can reduce the symptoms but not eradicate it. And these drugs have side effects, which both of these things are limitations. Additionally, we see how biotechnical treatments are starting to appear. Biotech companies are working tirelessly on a drug discovery for people suffering from this condition. Discoveries include deep brain stimulation using neurological biotech that uses implanted electrodes directed into a specific part of the brain that reduces the symptoms of Parkinson's. Sofamine is currently being tested that consists of dopamine created from human retinal pigment cells that are intended to be injected inside these neurons and begin to produce more dopamine. Discoveries like this are crucial. We also see how NIL-A, a small living molecule that works by initiating repair and growth in brain cells, has proven also to be effective. We also see how in our ever-present technological world that in the near future we could possibly find a cure, as biotech is becoming so advanced. The importance of funding research into this condition is fundamental and significant, as approximately 10 million people worldwide live with Parkinson's disease. We see how funding is crucial as no real cure has been found and diagnosing this condition has its limitations. The $5 million grant would prove valuable, allowing researchers and scientists to dramatically kickstart ways into finding a cure to better help people living with this condition every day as they suffer so much in pain. This money would allow biotech companies to further excel their research into an antidote. This disease, comparably to other genetic diseases, needs the grant the most, as there is a multitude of people suffering from it. You cannot put a monetary value on the millions of lives this could help and change. I put to you, you will not regret this movement. Let's do this together and put an end to Parkinson's. Thank you very much.